afternoon, everybody. At first, I want to introduce myself before you. I am Dr. Chittabrata, Professor, Department of Mathematics, SKGI. And today I will act as the moderator of this session. And I want to express my warm welcome to our esteemed resource person, then the delegates, above all the participants of various reputed institutes and the participants from our institute. You know that this is the 6-day auto-level degree program titled Artificial Intelligence for Healthcare Excellence, Innovations, Challenges and Future Trends. And today is the 3rd day, and now we are going to start the 6th session. So, before going to start the session, I want to introduce our resource person first. The name of our resource person today, you know, Dr. Chittabrata, who is currently serving as an associate professor in the Department of Bioinformatics at MAKAUT, West Bengal. Prior to this, he worked as an assistant professor at MIT University, Kolkata and MAKAUT. He earned his PhD in bioinformatics from Biophysics, Molecular Biology and Bioinformatics Department, Calcutta University in 2015. Following his BSc and MSc in biotechnologies. During his early research career, he was engaged as a research intern at the National Institute of Biomedical Genomics for 1 year. During his PhD, he has awarded prestigious fellowship including the BST Pass and UGC RF SMS. Dr. Noll has authored around 30 publications, including high-impact, peer-reviewed research articles and book chapters. He is an active reviewer for reputed journals such as Scientific Reports, BMC Genomics, Gene and Discover Oncology. Additionally, he serves as an associate editor for journals like Plant Gene, Human Gene and IJBST. He is an active researcher in the field of bioinformatics with a strong focus on integrated omics, computational biology and healthcare applications. So, it's a great pleasure to all of us that today we have got such an eminent personality as our resource person for this session. And before going to start the session, we want to felicitate our speaker who will present his views and ideas on the topic transformative role of artificial intelligence in the telehealth domain. So, before going to start the session, we want to felicitate him. For that, I am welcoming, or I want to invite first Professor Dr. to come up on the dais. Then Professor Tipti Gundishas, Principal SKGI to come upon the dais. Next, our coordinator of the FDP, Dr. Moshmi Adha Madam. So please come, Madam, here. Now I want to request Mr. Shubhi Kumar Ghosh from Accounts Department to come here, please, and Mr. Vinay Chaudhary from Maintenance Department. Please come here, Vinay Babu. Now I request our esteemed guest, Professor Dr. Chittabrata to come here, please, sir. Now I request Mr. Shubhi Kumar Ghosh to felicitate our resource person with the rose. Thank you, Shubhi Babu. Now I request Mr. Vinay Chaudhary to felicitate our resource person with the bag. Thank you, Vinay Babu. Now I request our Principal Sir Dhruvashi Sarkar Sir, Principal Madam, Tripti Madam, and Moshmi Adha Madam to come here and to present the memento to our resource person, Professor Dr. Mal. So thank you, everybody. Okay, now without wasting time, we want to start our next session entitled Transformative Role of Artificial Intelligence in the Telehealth Domain, and I request our resource person, Dr. Chittabrata, to start his session. So please come, sir.
and it can reduce error margins due to high precision because they are using robotic hands. AI can learn from thousands of surgeries to assist and guide in the rare or complex cases. For example, Mediterranean system, you can see the picture of this. This is the system. It is using AI-driven analytics during surgery. Now, predictive healthcare using genome plus health data. telehealth through automation, faster diagnosis and remote access. While it offers immense potential challenges like ethics, bias and privacy, need careful handling. The sector is rapidly growing, offering exciting career and research opportunities. With this note, thank you for attention.
Now, if you have any question on this topic, you may ask the question for your clarification to our side.
So if there is no question, so then I can ask one question. So that is whether you will choose AI for your need or not. Yes or no? Sorry?
For assistance or support?
For assistance or support. Yes.
In a controlled manner.
In a control.
But depends on the work.
Depending on the situation. Right. Depending on the situation, this is very critical. Means when you will use or when you will not use. Okay, fine. But it is required.
Yes.
Ultimately it is required because after COVID we all face this challenge.
Sir, when you slide, there is the one point you were mentioning that the face expression will detect the. So in our healthcare management, there is a called nursing assessment. So when patient comes inside the hospital, so emergency team or nursing people have to do the assessment of the physical one-to-one parameters. So in this, there one is the point is called body scale, means face expression, the pain scoring. So is there any possibility if we convert that one whole things to the AI? So if they take the photo and the pain will automatically means they can score according to the doctor can
Sure.
specify.
Sure. That is a good proposal. You can do that. I think there is no problem.
That will be the partition means the
Right.
accuracy
But,
of
but you need to get multiple of samples. You cannot do it in 10 samples or 100 samples. You need thousands of samples. So then only you will practice. Because nowadays you have seen the Google Lens, right? Google Lens. If you have used that thing, if you just click on the mobile Google Lens, so it will detect. But how it is detected? They are actually behind the scenes. They are analyzing multiple samples. That's why they are.
for that one we need huge data of the patient face expressions.
Every time, even if you are doing some simple calculation, then also you need multiple samples. In next level, I am going to talk on that.
But that can be possible.
Yes. Because this is required. Ultimately,
That will reduce the time of the assessment. No? So, one nurse takes the another 10 15 days for the assessing the patient. So if that is reduced, then they will be converted to their work.
so that kind of integrity or that kind of implementation is required to apply that. And that's why we need multiple people from multiple sectors, interdisciplinary. Very good.
Any other question from anybody else? So I think no question is there. So before concluding the session, I want to say that we have learned many things by this session. That is how AI supports telehealth. What is the application of AI in telehealth? Then what are the recent developments? What are the strengths and weakness of AI in telehealth? Then what is the market value and job prospect in AI and telehealth? Then what are the future scopes and challenges to address? And what skills required to be an AI expert in telehealth? So I think you have enlightened and enriched with this session, and our sir already has given his ideas and views to you in a descriptive way. So I want to express my heartiest thanks to our sir, Dr. Chittabrata, and by this I want to conclude this session. Thank you very much. Thank you, sir. Sir, next session টা দুটো শুরু করুন, এখন start করুন। হ্যাঁ? Okay. We don't want to waste any time more for the next session. So I think that it is not necessary to introduce again or share for the next session. And for that, directly I am going to the next session, and the session is on the research methodologies in bioinformatics and healthcare. And on this topic, our sir will give his ideas and views. So, sir, you can start your next.
Thank you, sir. So, I think we can start, right? We don't need break. Okay. So I'm going to talk about the research methodologies in bioinformatics and healthcare. So I have divided the session into up from healthcare sector. So now we are going to check what is healthcare. So you will agree with me that healthcare refers to the organized provision of medical, preventive, diagnostic and therapeutic services to maintain and improve our health. Now it encompasses a wide range of services from the primary care to specialized treatment including mental, physical and other. And it can be delivered through hospital, clinics, telemedicine, community programs, etc. Now modern healthcare integrates this technology, data analytics and personalized medicine to enhance outcomes and accessibility. Now bioinformatics will fall into which category in this healthcare definitions or objectives? What do you think? These are the preliminary or basic outline of healthcare. Sorry. Right. Not related. Because in hospitals or clinic, telemedicine, whether bioinformatics directly involved into this? Related. Indirectly. And most notably, bioinformatics falls into this because it can help to technology development, to data analytics, or it can be useful for personalized medicine. So that is the role of bioinformatics in healthcare. And we are going to explore different fields which are nowadays actually helpful for this.
Sir, is it related to drug discovery?
Yeah, of course. It's a huge area of drug discovery. Right. Because drug is required for healthcare. Now, this is a new topic, that is omics. Omics in healthcare. Now, so as you have mentioned that drug discovery. So drug discovery, how you will discover the drug? Any idea? explore the tools, different tools, AI-assisted tool or general tools, freely available tools, digital tools that can save time and reveal emerging research themes and gaps. You can explore websites or influencer journals. Try to visit the websites of high-impact journals in your choice of field. Look for the sections like key concepts or research highlights curated by the experts. Review the references list of highly cited papers to trace foundational or emerging ideas. Identify frequently cited authors or topics to understand current trends and gaps. This is very important because whenever you are trying to find out the literature, you see that how much time that literature is already cited. Suppose you are working on a drug, and that drug, two drugs I can say, and check two drugs for same disease. Now which drug you will use if you are trying to think in this manner? So you check the citation number, and in how much time that much citation about. Citation means when you are working on some research, so reference. So you check that number and check that in how many days or years that kind of reference number has been achieved or citation has been achieved for a particular drug. So a drug has been achieved 3,000 citations within a single year, and a drug has achieved 200 citations in 10 years. So you will surely keep your mind that you will go for the first one. So within short time, why many researchers are using that? So there is something. So try to explore that area. And every time make a note of your research. Record any questions or doubts that arise while reading the literature. Always link your questions to the source or prevent accidental plagiarism also. Use table diagram digital tools to organize your thoughts. And this practice helps in setting raw ideas into well-defined research problems. That means when you are going to design a project or research, try to use some documentation from the very scratch. That will help you in later. Next is research each question. Check if similar questions have already been explored. Review the literature to avoid duplications. Because many times when we do research, often we are thinking that okay, this is a novel idea to me, and I can go for that. Without doing any literature review, just by thinking that it's a novel idea to me, and I try to do some research. This may happen to anyone, any research scholar, because after a few days or few years, when you will do the literature review more on that topic, you will find that somebody has done already that. So your time is lost. So before doing that, you should take consideration into this thing that do your research literature review very well. So in bioinformatics, you can say 60% of time we need to do literature review. Okay. And then we need to consider the practical constraints as well, like time, funding, equipment, infrastructure, etc., etc. So aim for balanced research plan that is feasible and capable of producing publishable and fundable outcomes. So ultimate aim to publish, and from that we can generate the new idea so that our society can be benefit. So that is our ultimate goal of research. So keeping on this mind, we need to identify the research gaps. And there are certain tools, these are very much important. Beware of the literature gap. A research gap often reflects a gap in literature, and identifying such gaps help you to justify the novelty of necessity to your study. So expressions like, you can see, I don't know whether you can see or not. So expression like has or have not been. Many times in the literature you will find this kind of phrases. Is required or needed. The key question remains like this. It is getting the result. He or she is not getting that particular result, but either our boss is continuously saying that no, you are doing something wrong, repeat it, I need that result. So that is one kind of blockage in our mind to get the definite answer that we want. Try to get rid of this. Open your mind and try to accept. Try to accept all the things, whether it is wrong or right, that is questionable. But you need to focus on the methodology first, whether the method is right or wrong. If the methodology is sound and if it is right, so whatever results you are getting, so try to address that. You don't try to manipulate the result. Otherwise, it will be problem. If you try to twist something in your result to get your goal, that is not the exact goal. That will be falsified. Okay. So these are the 5 tips how you can enhance the chances of serendipitous discovery. So first is nurture a prepared mind that can recognize serendipitous opportunities. So open the mind so that something good can be discovered for the betterment. Pursue multidisciplinary and interdisciplinary research, which is nowadays very much in focus, and create opportunities for casual positions. Embrace failure and nurture culture of experimentation. So often we are very much angry with our failure. Okay. So we should not do that. We embrace our failure so that we can do something new. And develop a psychologically safe environment. So try to get rid of the negativity. Okay, so these are the basic things. Now, when we are trying to focus on the bioinformatics, data analysis, to get the healthcare, to get the benefit in the healthcare support, so we should know how the data are generated. Because in every field, if you go into every field, you should know the instruments very well. Otherwise, there will be some problem. So instruments are the source of raw biological data, which can ensure data interpretation and quality control. And these are essential for designing experiments, selecting analysis pipeline, and ensuring reproducibility. And understanding how the data is generated, that is equal to better analysis of the decision and higher research impact. So if you see that all of you see this instrument, right? Yes or no? What is the instrument? Microscope. What is this instrument? This one? This one? This is not centrifugal. This one? Any idea? So only all of us know that this is a microscope. But many different omics data nowadays, which are very much important to generate the personalized medicine, are coming from this kind of instruments. So these are the sequencing machines. This is microarray tools. This is mass spectrometry. Okay. So we need to know the instruments and the data, what they are generating. Otherwise, we cannot interpret the data or results getting out from these things. So these are the key instruments. So next generation sequencing, mass spectrometer, flow cytometry, microarray scanner, PET scanner, etc. So these are the very limited list. There are lots of instruments are there. I just wanted to say that next generation sequencing generates huge market nowadays, and it uses genomic or transcriptomic sequences. We can analyze our genome, whole genome, or exome, or RNAs by this kind of tools. Similarly, mass spectrometer is used for proteomic microarray scanner, we can identify the gene expression data or protein expression data. We can generate the expression profile. We know that how many genes we have, right? You know that? How many genes we have? Nowadays it is saying that 19,000 or 20,000. So these 20,000 genes we can profile at a time. What are the scenario right now in our world? We can profile our expression pattern of these 20,000. So that can be done by microarray scanner. That can be done by next generation sequencing, RNA sequencing as well. And there are are considering the replicates, sample replicates, you often will do some mistake. You need to avoid those things, treating technical replicates as biological replicates, pulling samples without proper labeling, including batch effects in experimental design. So pooling means what? Pooling means we are taking the samples from different and then mixing that sample. So that is the pooling of the samples. And often we ignore the batch effects in experimental design. This is a great mistake because we should know when or how we are conducting the experiment. Suppose we are collecting blood from 2 mouse samples, okay, in one day. Now, we have collected the samples and we will process one sample on that day and process another sample after 2 days. Are you getting my point? So one sample we are processing today when we are collecting, and one sample we are stored and processing later. So this will create the batch effect, because when you are comparing the 2 data sets, so then you will find that the conclusion you are getting, that may be biased. That may be biased in different aspects, like how and when, how long you have preserved that thing, where you have preserved that thing, and whether you have analyzed using the same instrument or not, whether you have analyzed the same precise chemical or not. So many questions will be there. So try to avoid that effect in experimental design. And so best practices are use both type when possible. So use technical replicates and biological replicates for inference. And apply randomization and blocking in experiment setup, document replicates details clearly in metadata. I am coming to the point of metadata. Now, next point is very much important: data backup and security. Data backup and security. This is also one aspect in methodology development, because many times, I don't know whether you have faced or not, that you are working on some project and you have generated some data. You have stored that data in your hard drive or computer or whatever, in USB. Now, when you need this data in emergency, I can say, at that time, often you will find that the data is not there. That is corrupted. That USB is corrupted. During my research work, I can explain that during my research work, when I was writing my dissertation, or not dissertation, my thesis, PhD thesis, after 2 weeks of work, when I tried to use my USB port, that is not working. So my whole data, 2 weeks work. So that's why data backup and security is very much important. we are going to see that how this data we get as a researcher, and for that we need to go to different website or web server, you can say, databases. So scientists who are working on this field, they are generating data, they are uploading their data to a server, and we are downloading the data on that server. So these are the different databases where you will find different types of data, like genomic data from NCBI and COTCGA project, proteomic data from Bright, HMDB data, clinical health, maybe ICU, Synthetica, etc. and EHR whatever data, after health kit, Fitbit data sets, etc. And there are multiple tools or platforms available to analyze this data, and we should know any one of these languages like R, Python, or we can use different platforms when we are not accustomed to different programming languages. So then we can go for the pipelines which are meant to analyze different kinds of data. They have already built some pipelines. We only click and we can analyze the data using Galaxy or Tabarn or K9. These are the tools. And these are the resources. So you all know that these are the different platforms where you can do some courses on this kind of data analysis. And what is the importance of metadata? I have already mentioned that when we are trying to get into the data, so then we need to know that where from the data is coming, how we are generating the data, what are the different parameters set during the experiment. So that kind of information, relevant information with that data is known as metadata. Okay. And you can see that this is the NIH portal. So SRA database portal, I have just draw a cartoon here, got a cartoon here. So you can search for any kind of data here, and if you search it, they will give you samples of data for download. And you can see that if you search this project here, you will get this kind of information. So this is the information and this is the metadata, you can say. So how the experiment was designed, who submitted the data, and how, on which organism or on which instrument these are generated. So all kind of information are there. And this is the data link where from you can download the data. Okay, these are long data, probably this kind of sequencing data, 1 GB, 2 GB, 3 GB data you need to download and work. So this is time consuming as well. And after that you need to analyze it. So if you come to the next part, that is the data collection and now data quality assurance, data quality check. So quality control. QC means quality control. It requires to remove the biases, errors in the data. And there are different QC tools. If we are considering the sequencing data, so this tool is very important. Fast QC tool. And fast QC tool, you will get this kind of figure by which you can analyze the data quality. You see, this is the before quality checking, quality trimming, you can say, and this is the after quality trimming. So to know this, we should know what is sequencing, then how this kind of data are generated. So I am not going into the details of this, but we can see that using this kind of tool, we can separate out that which data are good and which data are bad. So we can proceed with the good data set for further research. So these are the pre-processing techniques. So when we are downloading the data, we need to do the trimming thing. We need to apply some filter. We tools are there for doing these things, and different statistical tests are also there, because statistical corrections are required for getting to remove the noises from that data. Now, I would like to mention that this is the process. I showed you the microarray data set, microarray instrument, and from that instrument, this kind of raw data is generated. So this is the image file basically. Basically, this is an image file, and from that image, it is converted to an matrix, expression matrix, gene expression matrix, and these kind of experiments can say take one day to generate the data, and this is huge data. Earlier days, if you go back in 2005, 2010, so then most of the data you will find that they were used microarray data. So then the transcriptomic data, that is the RNA based sequence analysis done on this microarray. But after that, science evolved and we came to that NGS data. NGS means next generation sequencing. So next generation sequencing techniques, using next generation sequencing techniques, we try to generate more in-depth sequencing. So data volume also increased several folds. So if it gave us some MB size data, one data set equivalent to 20 MB or 30 MB or 50 MB max, this NGS data generates around 1 GB or 2 GB data. So you can understand the sequencing depth increased manifold, and that's why the data volume is also more. And to analyze this kind of 1 GB or 2 GB data in this kind of ordinary computer or laptop is not feasible, right? We need infrastructure to develop, and for that we can use this kind of tool. So this is very useful tool, this Galaxy, and this Galaxy standard data format. FASTA is very much known format, FASTA format, and here this is FASTQ format. Okay, and data quality provenance, that is quality control is to check by FASTQC or multi-QC protocol, and data backup security I have already mentioned. Data submission sharing is also repeated because from that public repository only we can get the data, right? And okay, so all of these are already mentioned. Now one thing we should mention that we should protect our privacy, data privacy law, and we should maintain the fair principle. I think in AI driven data FTP, we have already understood that what we mean by data privacy. I am not going to discuss about it in detail, but you should maintain the fair principle. That is, four principle is very much important for data oriented work. That is findable, it must be findable, it must be accessible, it must be interoperable and reusable. Often you will get some data in through some web browser that the data is modified or data is not accessible after some minute. So don't rely on that kind of data. Try to use some authentic, reliable data. Every time when you are searching, when you are developing your research methodology, keep in mind that your database should be authentic, reliable, which must be reusable. So many good, what I say, databases are following fair guidelines nowadays, so that it can be findable, accessible, interoperable, and reusable. And these are few topics which are focused on AI in healthcare. So digital biomarker identification from wearables, single cell omics and spatial transcriptomics, AI in drug repurposing and target prediction, and precision public health using EHR mind. Challenges and pitfalls, I have already mentioned some of this. We should minimize the batch effect in omics data, and if we are using some machine learning models, we should not overfit the model. We should check the data leakage and improper splitting of the data, and ethical approval will also be monitored. So, to summarize, bioinformatics research is systematic and interdisciplinary. Begin with good questions, ensure quality data. Sound methodology is equivalent to credible and reproducible results. Use updated with emerging tool and trends. Integration of bioinformatics and clinical informatics accelerate discovery, interpret results clearly, and publish effectively. That's it. Thank you. So this is the acknowledgement slide. I must thank our HOD and director of our department at MAKAUT. And these are the students who are working with us. I was known as Subodhip, and they are first in the last couple of years and currently placed in different university institutes. And these are the collaborators from Boston University. And very recently we have published the paper with these students, and they have also placed in different centers or institute. And the main thing is that they are working in their domain. Many times we have seen that they are studying biotechnology but working in dam kind of thing. So they have worked in biotechnology or bioinformatics related thing, and they are working in that domain. So that is the good thing, right? And I must thank my guide, Professor Sundu, and organizers of this seminar, and especially Dr. Devashish sir, and to all my teachers, friends and family. So last thing, do innovative research and take challenge. Thank you.
So if you have any question in your mind, so you ask the question to yourself and he will clarify all the questions. Any question from anybody?
Sir, may I ask you what is the minimum computational time required to in your papers?
So this model I have shown.
This model or this paper that is related to yes, cancer treatment or
That depends on your configuration. So we have used probably 24 GB RAM machine, 24 GB RAM machine only, and it took 50 minutes to run each one. Nothing else. Very small set of data.
that means whatever the input I am giving after 15 minutes the machine will give the result. That is the minimum.
Configuration or
Means we have used very small set of data. That's why it is taking less time.
Okay.
If you are using lots of data, so accordingly the time will require.
So 15 minutes is too much.
Sir, machine learning we run some of the algorithms are even that is not sufficient to train the model.
It is nothing.
No sir, to say in the real time operation we have
No, no.
So you want to
It is not going to real time, na? It is just predicting some biomolecule. So after that, you will design something else. So it is.
That is training time, not prediction.
So once the training is being done, after that it will give you result fast. But to train the model, it's similar the learning process of a human.
so what is the testing time sir?
No, I mean training and testing both. We need around 15 to 20 minutes for each.
It's small amount.
Small data. Small data.
It's not
Small data.
data size is large and the model is complex.
Sir, with GPU configuration or without GPU?
Without.
Few days
Simple.
depends on that. জিনোমের কিছু প্রজেক্ট মেটা স্টার্ট করেছে কয়েক বছর আগে এখন চলছে
As you are using very small data, is it not prone to overfit?
No, we have checked the data is not over.
But
So that's why we have taken it into different sets. You can see that we are using the data into subset 1, subset 2 and subset 3.
what is the difference between the validation and the testing and this?
The difference in terms of
Difference means both the accuracy it is showing for validation and the testing.
right.
Is there any
So
difference?
it's about 1 means 0.98, 0.97. So if you go to the paper, I don't remember the exact value, but it's almost 0.96, 0.98.
Yes, any more question? If no, so I think that you have got enough knowledge and enriched, enlightened by this session through his presentation on the topic research methodologies in bioinformatics and healthcare. And I also think that this session provides the participants with a foundational understanding of contemporary research paradigm, essential for impactful scientific contributions. It covered robust strategies of data collection, comprehensive data analysis, and effective methods for extracting actionable insights from large-scale, complex biological and healthcare data. I think that the participants will explore cutting-edge research methodologies that are revolutionizing bioinformatics and healthcare. Special emphasis were placed on the role of high throughput omics, that is omics technologies, in biomarker discovery and personalized medicine. In addition, the session divided into the integration of artificial intelligence and machine learning techniques for processing vast biological and clinical data sets. And he also gave details starting from the pre-processing techniques to post-processing techniques, then bioinformation pipelines, what are the tools and best practices, current trends, methodologies, and innovations, what cautions we will face, then how to choose the right journal, what research methodology we have to follow, what are the different types of omics in health, like genomics, 3D genomics, then epigenomics, transcriptomics, then proteomics, metabolomics, etc. And what are the omics in disease? What are the scientific methods and hypothesis design? So many topics from the statistics also covered in his presentation. We have seen like data quality, what are the pitfalls, what are the data collections, what are the metadata, then pre-processing the data, post-processing the data, then the methodologies, innovations, then data interpretation, representation, then what are the checklists. So, etc. In this way, we have seen many things from the statistics as well as from the biostatistics he has covered. So, thank you very much, sir, for your fantastic presentation and giving details about your topic, research methodologies in bioinformatics and healthcare. So, thank you very much, sir, for your presentation. So, now I request Professor Devashish that to the stage, and also Boshi Madam is come here to present the presentation certificate to our sir for his fantastic presentation.
But I must say that this might be boring to many different field people, because bioinformatics is generally boring. Okay, I must take that note, because I feel that during my class audience also, students are also feel bored. But after a few days, they will be accustomed, and after that they show their capabilities in this. So that is the good side.
We have also stimulus foundation as like I am proudly presenting this certificate to you. This is an off the record. If you take out the promotion and other criteria from the research, I think 0.01% of the people will do research here. Research itself is boring. There are very few minds in the world who do research generously. But that session I have seen, such kind of presentation I had before doing the PhD, I could basically limit my PhD duration. So in that way, you have taken a great presentation. I'm sure everybody will agree. Thank you.
Madam,